How good do biomarkers have to be to make them useful? So to address this question, I'm going to tell you the story of what we've done in our team on early lung cancer detection biomarkers, because we think a lot about how good do they have to be to make them useful. And I'm going to try to explain what the principles that we work under and how we apply them. All of this work was led jointly with Matthias Johansson at I-ELCAP, who co-leads my team. He's from Sweden. So as I alluded to before, the sort of core thing that we are really worried about in this field is study design. And the sad fact is that most early cancer detection biomarkers ultimately fail to translate to any patient benefit whatsoever. This is not just for lung cancer, but this is for early cancer detection in general. In my view, the key reason, not the only reason, is poor study design. So we have sort of a few principles by which we organize our own research. The first is to use a sufficient sample size. So don't scan 5,000 markers with 20 cases. You're not going to succeed. The second is to represent the intended use population. If your cases are overwhelmingly people with a smoking history and your controls are 90% people without a smoking history, you're going to identify biomarkers of smoking, but that's not what you want to do. Third, use independent discovery and validation. You will always find things in your discovery. You will manage to develop an algorithm. Does it validate? Does it perform well in an independent sample is the question. The fourth is to demonstrate utility in pre-diagnostic samples, because this gives you a proof of principle that the biomarker could have found the cancer earlier than it was found in real life. And finally, to demonstrate an improvement over already available information. So if we're going to stick a needle in somebody's arm and we're going to pay for a test, we should be able to show that we're going to do better in terms of prediction than we were going to do with what we already knew. So for pre-screening settings, what this means is doing better than a sort of well-performing risk prediction model like the PLCO model. In post-screening settings, this means adding information to what we learned from the LDCT image, including when you apply some sort of AI to that image. We need to do better. So we have to have a benchmark. I'm going to skip this. Okay. The story I'm going to tell you used a specific type of study resource, and it's important that you understand precisely what it is, so I'm going to walk through it. So we work primarily in population epidemiological cohorts, and these are groups of sort of generally healthy, cancer-free people that are walking around in their normal lives. These cohorts typically enroll between, say, 10,000 and 500,000 people. The participants fill out detailed questionnaires at enrollment on risk factors, health behaviors, and so forth. And then they often donate blood and other biospecimens, which are stored at -80 Celsius. And then they're followed over time to see what happens to them. So all types of cancers are ascertained, usually by linkage to registries. Mortality is ascertained in great detail most of the time with corresponding dates and so forth. So then you can use these studies to perform many different types of research. In my team, we coordinate the Lung Cancer Cohort Consortium. So this is a consortium of 25 of these population epidemiological cohorts from around the world. Taken all together, we have a harmonized database for the LC3 of 3 million cohort participants, among whom about 69,000 lung cancer cases developed during the follow-up. And this is really the engine that's driving all of our research on risk prediction models, on biomarkers, on epidemiology, and so forth. We finally managed to have a website for the consortium, which is listed at the bottom here. And we do have a data sharing setup for non-commercial research. So I would encourage you to check it out and reach out if you're interested. But for the biomarker work, what we're doing in these protein biomarker studies is we restrict to participants with a current or former smoking history. That's what we've done for the moment. People who could be considered for lung cancer screening. And for the lung cancer cases, we select those that developed within 3 years after the blood draw. So we're really looking for biomarkers that are providing information before diagnosis, but are sort of short-term early detection markers. So you see already these sort of two principles playing out. We're trying to represent the intended use population, people with a smoking history who might be considered for screening, and we're trying to demonstrate utility in pre-diagnostic samples. All of this work is done within this Integral U19 program that Chris Amos leads. Stands for Integrative Analysis of Lung Cancer Etiology and Risk. Chris is leading Project 1, which is focused on genetics. Matthias and I are leading Project 2, which is focused on risk biomarkers. And Project 3 is focused on nodule malignancy biomarkers and radiomics. And this is led by Regine Hung in Toronto. A number of you are working with her in this context. And so together with Regine, Project 2 and Project 3 combined had a goal to develop and validate a protein biomarker panel for early lung cancer detection. And we chose sort of a priori to focus on proteins because at the time they had really the best evidence that they could provide more information about future lung cancer beyond risk prediction models. Integral was funded initially in 2018. So this is when we set up this biomarker part of the consortium. And then during 2019 and 2020, we ran a multi-phase discovery analysis. So we used assays from Olink Proteomics, which is now quite a well-known global company, but at the time was fairly small. And they started up in Sweden. And back in 2019, 1,200 proteins was the most you could measure. Now it's many more on these individual 96-plex panels. So we scanned all of the proteins that we could using these Olink assays and then did sort of a replication phase. These are the results of our discovery protein work. These are published, and you can pull up the publication and sort of look at this in detail. But to make a very long story short, we analyzed 731 case control pairs selected from 6 of the cohorts in the consortium. And we identified 36 proteins that were robustly associated with risk of lung cancer developing over the next 3 years after blood draw. But this is only half of the story about this protein panel. So in parallel, Regine in Toronto was working with CT screening studies, including a few of you in the room, and she selected samples from participants with malignant nodules, as well as participants with benign nodules. She conducted a parallel study scanning the same proteins, and she identified proteins that were indicative of nodule malignancy compared with benign. So we were able to put up to 21 proteins on a customized panel with absolute quantification. So on each side, we sort of made our protein wish list, and we put these together. And at the end, essentially, there were 7 markers identified only as risk markers by us in the cohorts. There were 7 markers identified only by Regine for nodule malignancy, and there were 7 markers that overlapped. So we put these markers onto a panel using the same Olink technology, but now with absolute quantification. This was happening basically in 2021, and then there was a phase of sort of technical development of the panel. And then we ran a very large number of assays for a validation phase, which would be split between training and testing. I'll tell you about that in a second. In 2023, the second round of Integral was funded on the first submission, which was excellent. And we carried out the statistical analyses to train the algorithm, incorporating the proteins. We locked down the algorithm and then unlocked the testing set of the validation data and validated the model at the end of last year. So I'm able to show you these results now. As I mentioned, this consortium of cohorts is really the engine driving all of this, and it allows us to design this research from the very outset in a very robust way. So I showed you the results already from the discovery, which is on the left side. We scanned these 1,200 proteins. We used a matched case control design, which was nested within 6 cohorts. Then we moved to the validation, training and testing phases. And here we changed to a case cohort design, which if you've taken some epidemiology means that we can have a perfectly representative sample from the original cohorts when applying statistical inverse probability weights. The training phase included 7 cohorts with a total of about 2,000 participants, among whom about 800 developed lung cancer within 3 years after the blood draw. And then the testing data, which is the part that was locked up, had a completely independent set of 7 cohorts with about 1,700 participants, including about 560 cases. So I'm going to show you some results, and these are from the independent testing phase. And here again, you can see these principles sort of playing out. use a sufficient sample size. This requires a lot of money and a lot of planning, but that's what we did. Use independent discovery and validation. And this is the power of the consortium, because we can divide up these cohorts across the phases so that we have complete independence at the end. So I'll show you here the validation results from the testing phase. What we did is, using the training half of the data, we trained a prediction model for risk of lung cancer over 3 years, which can be modified to predict risk over any time period between 0 and 3 years. In this model, we accounted for age of the participant, the number of cigarettes smoked per day, the smoking duration, and then the absolute quantification, the absolute concentration of 13 proteins on the panel. So we don't need all 21, but we could add information all the way up to 13. We then wrote a protocol in which we specified in gory detail the parameters of this model, and in gory detail, the procedure by which we would evaluate its performance. And we posted this on a protocol server to keep ourselves honest. So here on the right side is an AUC curve predicting lung cancer up to 1 year after blood draw. So first, again, we need a benchmark, yeah? So we have the PLCO M2012 model, which had an AUC of 0.79 in our sample. This is exactly what we would expect. And when we looked at the performance of the protein algorithm, we got quite a large increase in the AUC, all the way up to 0.88. And when we got this result, we were really sort of jumping up and down, because when you have forced yourself to be completely honest about not tweaking your algorithm, and you have locked down your data, and you have no wiggle room for how you're going to evaluate it, you are very scared. And we were very scared, and we were very happy when the performance came out to be quite good. But really the reason is that we had such a large and diverse training sample that when we went to the testing, we didn't lose a lot of performance. So again, this is the benchmark. We demonstrate an improvement over the information that we already have, which is the PLCO model. This graph looks similar, but it's a bit different. So this is sort of trying to gauge the potential impact of using this type of biomarker in a clinical setting to decide who goes to screening and who doesn't. On the x-axis, you have the percent of people who go to screening. So the percentage of people with a smoking history who are eligible for LDCT. And on the y-axis, you have the percentage of lung cancer cases over the next year that you manage to catch that are in that screening population. So if we take the Task Force 2021 criteria as a benchmark, the Task Force 2021 criteria in this data screen 25% of people. And in those 25%, they catch 63% of lung cancers that are occurring over the next year. So already this is quite good performance for the Task Force criteria. If instead we still pick 25% of people, but we pick different people, and we use the PLCO model, we increase the percentage of cancers up to 70%. And finally, if we still pick 25% of people, but we use the biomarker algorithm, we get all the way to 85% of cancers. So this is sort of this increase in sensitivity that we can obtain using different tools all the way up to a biomarker. So let's say we meet all of these benchmarks. We design our study in the best possible way, and we sort of get to the end. How much improvement is enough? And I don't think there's one answer to this question. There's many answers, because the answer is it depends. It depends on the cost of the test. It depends on the ease of its use. And it depends on the magnitude and the nature of its clinical impact and the stage of the screening process at which it acts and what it manages to achieve. This question, I think, is really best addressed through, there's not a single answer. So you sort of have to model it, and it's best addressed through modeling. If the NCI doesn't cease to exist, then the integral panel that we've developed will be evaluated in the next round of CISNET as a sort of use case for blood biomarkers. However, this is not the end of the story. It's a really long road to do this early detection biomarker work. And we have many, many different things sort of funded and underway. The first is a feasibility and acceptability study, which we're doing with Mike Gieske in Kentucky. We are identifying now biomarkers for lung cancer among people who never smoked. I didn't have time to show you, but we have some really nice pilot data for that. And we're going to run probably up to 700 case control pairs. We are going to evaluate if we can improve prediction using repeated measurements. and also by integrating germline genetic information. So this is a concept called demendalization. The idea is that some of your protein levels are determined by your germline genetics, and if we remove that variation, then we can get improved prediction from the sort of biomarker cancer association being better defined. We are quantifying utility for nodule management in a number of studies together with Regine. And what we really want to do ultimately is a sort of large prospective interventional study, which in my mind would have a design sort of analogous to the International Lung Screening Trial, where we sort of screen everybody who's eligible based on either tool, and we make a Venn diagram at the end. So I want to say a huge thank you to my team. I don't have everyone here, but I think are currently 16 people from 13 different countries. This was an enormous amount of work over a long time period. Most of the analyses that I showed you were led by our postdoctoral fellow, Hannah Zahed. We also have an incredible network of collaborators around the world who contributed their samples, contributed their data, and contributed their scientific expertise as part of this collaboration. And I'm going to make a few very quick shameless announcements. We have 3 postdocs on the job market. The first one is Hannah, who led these analyses. The second one is Tina Onwuka from Nigeria, and the third is Ali Dominguez from the US. We're open to hiring postdocs if you have any PhD students graduating, assuming we don't lose all of our NCI funding. And most importantly, we're quite happy to collaborate on further studies with this biomarker panel. It's sort of an iterative process of planning ahead, producing kits and shipping samples and doing the measurements. So if you're interested, please come talk to me. All right, thanks.